So I'm going to tell you about what we're doing with Trinity software and how we've been making use of some of the supercomputing systems. Now, RNA-seq as a whole has been just a phenomenal technology, and that it enables you to study the gene expression and gene content of basically any living organism on the planet. You just need to be able to isolate the transcript sequences, convert them to cDNA, and then throw them onto your favorite next-gen sequencer. When you do sequencing of transcripts on something like Illumina, you're not necessarily going to get full-length transcripts, right? Usually you end up with reads that are much shorter than the transcripts that you're starting with. And so there's various challenges in making use of these data, including being able to reconstruct the transcripts from which the short read data were derived, and being able to estimate their expression levels. Now, there are a couple of different ways to do this. If you have a genome sequence, then you can map your reads onto the genome, and using a specialized short read alignment tool that will take into account introns found in the genomic sequence. And then given the alignments to the genome sequence, you can then reconstruct the transcripts. And there's this tool that many of you are probably aware of, the Tuxedo package that has Top Hat, which will do alignments. There's actually a dozen different tools that will do this, and a tool called Cufflinks that will reconstruct transcripts. But this requires that you have a genome, and a genome that's actually a good representation of where the RNA-seq reads were derived. But if you don't have a genome, there's still another option. Or if your genome is heavily rearranged or restructured, or it's just not really the best substrate for trying to analyze your data, you have another option, and that is just to assemble the reads directly, doing de novo transcriptome assembly. And we developed a tool that will do this called Trinity. I'm going to tell you a little bit about Trinity. Yeah, sure. Anytime, feel free to make this interactive. So.
If you go back a slide.
Yeah.
This approach is especially useful in things like analyzing tumor data, so cancer, because the cancer genome is particularly messed up. So you can't quite just align to the germline DNA. So.
All right. And that's a really important point. I'm glad you mentioned that. And towards the end of the talk, I'll mention some things about our applications in the area of cancer research. So we designed Trinity from the ground up a few years ago. It's probably been like 4 or 5 years ago now, to take into account the various characteristics of transcript data as part of its reconstruction. It takes into account the differences in the expression levels between transcripts. It takes into account the complexity of alternative splicing, so it'll reconstruct the different isoforms that exist within your data set. And it takes into account both sense and antisense transcription. So when you generate your RNA-seq data, you have an option where you can generate your data in a strand-specific way to retain that strand information. And that's really key if you want to be able to study things like antisense transcription, which is an important process in regulating transcription, regulating gene expression. Without that, you can still reconstruct transcripts, but it's not obvious. You don't know necessarily that it's coming from the antisense versus the sense, especially when you have long single exon genes. So we took all this information into account in building our software tool. We published it in 2011, and we actually made the cover of Nature Biotechnology. I really love this cover, and I thought I'd mention that it's the wallpaper. It's been the wallpaper on my cell phone since 2011. I just love it. It's such a beautiful image. Anyway, so we published it, and last year we followed it up with a Nature Protocols paper that shows, basically walks users through how you generate an assembly from RNA-seq data using Trinity, but more importantly, what do you do once you have an assembly, right? Because that's really a starting point for doing downstream analyses. So in this case, if you're starting with a bunch of different tissues, you want to generate an assembly in a genome-free way, and then you can do things like estimate your transcript expression levels in each of the different tissues, do differential expression analysis, generate various kinds of plots, as we have ties into Bioconductor and generating PDFs and generating various data sets that are useful for your investigations. Looking at expression clusters, correlation plots, and pulling out those genes that look most interesting in the context of your experiments. The software, documentation, It's all freely available. We put it on SourceForge as our website. The documentation is fairly extensive, and it basically walks you through how you do all of this, similarly to the protocol paper. We call it Trinity because there's a number of reasons why we call it Trinity. There's 3 different tools. There were 3 of us that were working on it. Each of us developed each of these different tools. Trinity sounded like a good name for it. Lots of 3s there. It starts with the Inchworm process, where we start with the RNA-seq reads, just the raw reads, and Inchworm does a very quick building of draft contigs, doing a greedy k-mer extension, and I'll describe in minute detail how that works. So we get these linear contig sequences from the reads. There's a process called Chrysalis, which comes in next, and what that does is it clusters together those contigs that are related to each other, such that they might represent different parts of alternatively spliced isoforms, or correspond to paralogous sequences that have shared sequence in common. And ultimately, ideally, we'll end up with 1 cluster of contigs per gene. And then it'll construct the De Bruijn graph. A De Bruijn graph is it's a data structure that's very commonly used in assembly, both genome assembly and transcriptome assembly. And how this works is basically every k-mer, so every length of, let's say, 25, every length of 25 in a read is extracted and forms a node in the graph. And those k-mers that overlap each other by all but 1 base are then linked together in this graph. And when you do this for all the contigs, you end up with a complex structure that might have loops and branches. And this basically represents all of the transcriptional complexity for that given gene. And you can reconstruct transcripts by basically just following a path within this graph. So to reconstruct the contig here, you might follow a path that goes like this. And then you have alternative paths where these bulges and branches represent different sequences that correspond to alternatively spliced isoforms or to different parts of your paralogous transcripts. One of the keys in reconstructing transcripts from these graphs is being able to follow the paths in the graph that are supported by the underlying read data. And that's what Butterfly does. Butterfly will take the De Bruijn graph, it'll take the original reads, it'll thread the reads through this graph structure, and report the final transcript sequences, the different isoforms as full length in many cases, and try to tease apart those transcripts that correspond to paralogous genes. As part of this process, like I said earlier, we end up with these clusters. Ideally, we'll have 1 cluster of transcripts and 1 graph per gene. Okay, and in that case, often it's the case that we'll end up with tens of thousands or hundreds of thousands of these individual graphs. But one of the beauties of that is that each of these graphs can be processed by a separate Butterfly process, and you can do all these in parallel, right? So you can imagine having 100,000 different assembly jobs going on in parallel, which makes it amenable to high performance computing, using grid computing, using other sorts of methods. I should mention also that since many of you are software engineers, you're probably very curious about how the code is wired together. 3 different people, 3 different code bases. C++, my code base, C++, Manfred Graber, his code base, and this is written in Java by Moran Yassor. And then we have a big Perl script that I wrote that basically glues everything together. So it's a combination of Perl, C++, we've got Java in there. There's a lot of R code for generating plots and doing various other kinds of analyses. So it's a hodgepodge of lots of things. And I'm sure it'll make a lot of software engineers cringe if they ever looked at it. But anyway, how well does it work? When we published it, of course, we showed everyone how great it was and how much better it was than other types of processes. And there's a lot of conflicting information in the literature. There'll be different papers that come out that say we're better than Trinity. And anyway, lots of different metrics that you can use for trying to compare these things and try to figure out who's better than which program is better than another one. But it's very gratifying when you see a third party publish a paper where they do an evaluation, and it's a very sophisticated evaluation, and our tool actually comes out as being on top. And this came out just a couple of weeks ago in the preprint archive. This work done by Bo Lee. And they came up with a couple different, very sophisticated metrics for evaluating how accurate an assembly is. So they have a couple of different metrics in here. They use mouse transcriptome data. They did this both with real data, they did it with simulated data. And I'm not going to get into this too much. It's pretty complicated. Mathematically, it's over my head, to be honest with you. Hopefully someday I'll understand it. Anyway, you save these two different scores. You have something called an R-semi-val score, which is it's a log joint probability of the assembly and the reads used to construct it. So it's good. It's a metric of how good the assembly is. And there's another metric for how good the assembly is, which is a measurement of the degree to which the assembly compresses all of the RNA-seq data. Right? So there are a couple of metrics, and they're highly correlated with each other. But in any case, you've got all these different tools here that have been developed over time for developing transcript reconstructions, and woohoo, we came out on best. So this is very gratifying to see that. This isn't just the one metric. There's lots of other metrics that they proposed. With another data set, which is axolotl, which is it's a salamander. It's a very commonly used for studies in limb regeneration. And it has no genome. The genome has no genome. Of course it has a genome. The genome hasn't been sequenced because the genome is huge. It's over 30 gigabases. But it's been a popular target for doing transcriptome studies. And they've been doing transcriptome studies there. We've also been doing it separately with a different collaboration. But anyway, as part of this work, they assembled the transcriptome of the regenerating limb tissue, and they compared the transcripts that they got out to the proteins from frog, right? Because the frog genome, Xenopus, has been sequenced, and there's a good frog protein data set. And basically, this plot indicates how well the reconstructed transcripts represent the full-length proteins from the frog, and doing a comparative analysis. And I mean, the key point here is, Trinity's came out best, so that's great. And we've got the highest score as far as their scoring scheme. So this was it was very gratifying, and I couldn't be happier when I was reading the paper. There's a couple remarks like Trinity produced the most accurate assemblies on all data sets, and varying the parameters of Trinity had a relatively small effect on the accuracy of the resulting assemblies as compared to some of the other tools, which produce assemblies that spanned a large range of accuracies. So that's all I'll say. Very happy about that. Many of you have seen this. This is a slide that many of us have shown over the years, and this just reflects the reality that we're in right now. It's insane how much the cost of sequencing has dropped over the recent years, and sequencing is essentially, think of it as being free, right? But it's not free. We know there's a lot of other costs involved in terms of hardware and compute time and storage, but the sequencing itself is free. And why? That's because these great improvements and the yields of the various sequencing technologies that we're using. And it creates some major challenges for those of us that are in this field and working on these data and developing software to try to tackle these different data types. And so what do we need? We need faster algorithms. We need to have better use of the hardware. And there are hard problems and there are easy problems. Coming up with more clever algorithms, that's a really hard problem. But there are some simple things you can do in order to try to tackle these data. And one of the simplest things you can try to do is just make it more parallel, right? Just try to make better use of the hardware that you have. Instead of funneling everything through one process, see if you can split it up and to 10,000 different processes and make it run 10,000 times faster, ideally, if you have good scalability. So there are certain parts of Trinity that are already very amenable to high-performance computing and using massively parallel computing. And I mentioned that earlier. It's because we have these steps that basically make tens of thousands or hundreds of thousands of these graphs, and each graph becomes basically a separate process. And an LSF or Slurm, we have basically support in Trinity for all of these different architectures for grid computing. And you can basically just fan them out and let them all run separately, and then when they come back, you just join all the results, and you're done. And that's great. But we do have, basically, we have a step where we haven't been able to do that. We have a step in Inchworm, which does the initial config building. It runs on one server. It runs in a serial fashion. It requires a lot of RAM, because it's going to take all of your read data and throw it all up into RAM. And it was a prime target for us to try to figure out better ways to do this, find some way to, one, make the algorithm work in parallel instead of serial, And then 2, experiment with ways of trying to distribute the computation and the data across other compute nodes. And that's really the main focus of what I'm going to describe today. Before I can dig into how we've done this, like how we've attempted to make it parallel, I'm first going to just give you a quick overview of how the Inchworm algorithm works. It's very, very simple, and you'll see how simple it is. We basically start with your reads, right? Take one read, take your first read, and we decompose it into the sets of overlapping k-mers. And you can see here, we just look at the first 3 k-mers. We've got these 3 k-mers here. They have an overlapping length. Basically, they overlap all but one base. This base is unique here, and this base extends here. It's basically just, just walk a little window, like every base, and just extract that k-mer. And you build a catalog of all of these k-mers. And we're not even retaining the information that this k-mer is linked to this k-mer. If you're building a proper de Bruijn graph, like I mentioned earlier, you'd actually you build a graph and you make this a node, you make this a node, and you basically say, okay, this k-mer comes after this k-mer by putting an edge between the nodes. But we're not even doing that, right? We're doing this even simpler. All we're doing is just pulling out the k-mers, and we're just going to build a table that has the k-mer and the count of how many times we've seen that k-mer in the entire read data set. This is what we're starting with. It's a hash table with key value pairs. Key is the k-mer, and the value is just the number of times that we've observed that k-mer in the entire read data set. And then from that, we start to build contigs. First thing we do is we prioritize the k-mers, and you'll see why we do that later. Yeah.
What is K?
What is K? In our case, like when we run Trinity, we keep it at 25.
25.
Yeah, so the overlap size is 24. So, K is actually 24, but the words we're extracting are 25 mers. But for this example, I'm just going to keep it simple. I'm going to use I use a Gattaca, right? It was one of our favorite k-mers. So they made a movie after it and everything, right? It's famous. So we want to take a seed k-mer. We want to start building the contig, but we want a place to start building that contig from. And in order to do that, we just take the k-mer, which is found the highest abundance in the entire data set. And there are certain requirements. We don't just take any top k-mer. We have to make sure that it has reasonable complexity. So we're not going to take the string of all A's or that you'd typically find in an RNA-seq data set for like poly 8 tails. So we just take something that looks reasonably complex. And we start with this as our seed. In this simple example, we're starting with Gattaca. We found that it's found in our data set 9 times. In real data sets, you've got it thousands of times or tens of thousands or more. And we want to extend it. So what we'll do is we'll start extending it to the right. And when we do that, we have 4 possible extensions. That's because our nucleotide alphabet is very limited, right? We only have 4 different characters. So there's only 4 possible extensions. And we want to choose the extension that has the highest abundance, doing this greedy search. So what we do is we look up the next possible k-mers, right? The next k-mer has to start with this A, and has to end with one of these 4 bases. So we just look up each one of these in our big catalog. We find that this k-mer is found 4 times. This was found once. This k-mer wasn't found at all, so it's missing from our data set. And this one's found 3 times. And we see, okay, well, the one with the G is found the most, so we're just going to take that. And that's the base that we're going to use to extend our growing contig. And we just throw out the other options, right? And we just keep doing this, right? We just go on to the next one. The next k-mer is going to start with a T, and it's going to end with one of these 4. We look up the possible ones, and we find there's only these 2 k-mers that actually exist in our data set. A is found the most, so then we're going to choose that, and we're going to extend.
So you have the expected number of times you would see the 24-mer.
Yeah.
In the transcriptome as opposed to the genome.
It's highly unique.
Is very low.
Yes, that's right. Yeah. Yeah. So why wouldn't you do something simple like this with genomic data? It's because of all the repetitive DNA that you're going to have, right? It just wouldn't. You still end up with nice contigs for like the unique regions of the genome, but as far as the genome assembly, it would be ridiculous. But this actually works extremely well for transcriptome data, just due to the high level of the uniqueness of these k-mers within the transcript set. And this is just like the initial step. So this is just a way of trying to carve up our data and get initial contigs, and the rest of Trinity would take over and generate like the final contigs and deal with repeat structures and other sorts of things that we're not really going to get into today. Yeah. Ami.
You're looking only on one, just the next one. So if there is, for example, the variant, you have both A and T, then you might miss one of them because you just take the best. You are not looking like, can you do two
Yeah.
steps ahead and then you can actually.
Well, you could. I mean, at any time you're going to end up with a branch point. I mean, you're always going to. Oops. Wrong button. There we go. All right. I'll try not to push that button again. All right. So anytime you're extending, you always have these options, right? And in the case where you have a SNP, either option could work. If there's a tie, then we try to break ties, doing a little bit of recursion. Basically you can choose one or the other. When I say we're not going to take this, we're not actually throwing it away. We're not discarding it. All right, so this will still show up later on as part of a different config. So we're still going to capture the bits that correspond to the variants, but they'll be in shorter configs. And then later on, we'll hopefully cluster the shorter config with the larger config in the crystal stage, build the de Bruijn graph that contains the variant information as a bulge, and we can decide how do we want to work with it at that point. Yeah?
Since you're assembling at the kernel level and you're decomposed to reads, isn't there a possibility that you are joining two pieces of two reads together? Like for instance, one piece from one bunch of tables from one read, like mix and match reads. Is that common or it's just?
It's rare, but it can happen. And that's something that would get solved in the de Bruijn graph stage by the Butterfly software. Yeah. So a lot of these things we thought about, but and you're right. These are the kinds of caveats, and that's why you don't just use this, right? This isn't the final result, right? This is just sort of like our initial way of trying to partition up all the read data so we have like a massively parallel computing problem that we can deal with. Yeah. But it does actually turn out to be useful because you still end up getting full transcripts, dominant isoforms reconstructed for most of them, just using this simple strategy. But you have to think about the alternative splice variants. You have to think about the cases where you're forming chimeras between unrelated transcripts, and that's sort of solved later on in Trinity.
Does it sense to the initial selection of the seed chimer?
Is it? Yes, it is. Absolutely. Yeah. And that's actually a key point that I'm going to address in just a little bit. Yeah.
What was the question?
The question was, is it important which seed chimer you use? And as I mentioned before, basically the seed that we chose as part of our serial algorithm is basically just taking the one that occurs the most, right? So it's like that's our prioritization scheme. And the idea there is that you'll end up selecting a chimer that is it's a high quality chimer and doesn't have errors and other artifacts.
So do you do it in both directions?
Yep. You're going too fast for me. We're getting there. Now we get the. Okay, so we keep going, and basically we just keep extending to the right until we can't extend anymore, right? There's no chimer in our data set that allows for that single base extension. Then we stop, right? Then we're done extending to the right. And as Ami figured out, we're probably going to want to do this to the left too. And that's exactly what we do, right? Once we're done extending to the right, then we turn around and we keep this part and we start working our way in this direction and just keep going until we can't go anymore. And then we've got a config, and that config is considered, the final Inchworm config for those chimers. We report that config as part of the Inchworm output, and then we go through and we take those chimers out of our catalog. Okay, so the catalog is going to keep getting smaller, and those chimers can't be reused, right? So anytime you find a chimer within an Inchworm config, it's unique. You're not going to find it as part of any other Inchworm config. So remove the chimers that correspond to this config, and then we basically cycle through and do it again. Pick the next highest abundant seed, and then do the extensions. Yeah.
Do you get bubbles as well? Do you cut those?
Oh yeah. So if you have a circular situation, yeah, we avoid that in the code. So if you're extending, once you've used that, you basically have like a local hash that keeps track of what's been used. Because otherwise you keep cycling through, and and that's happened like in the early development of the code. Yeah, that was a quick thing to solve, right? Yeah. All right, so this whole process can be described in just a few lines of pseudocode. We basically divide it into two processes. In the first process, we're just trying to get all the data into RAM. in the form of these k-mers in our k-mer catalog. So we're reading all the sequences from the files. We build this big hash table. And then we sort the k-mers by decreasing count. That's the way of building our prioritization of the k-mers. And then in the second part, after we've done that, then we're just going to build the contigs. And so while we have k-mers, we just select our seed as the highest k-mer that hasn't been used yet. We build the contig from that, and then we remove those contigs. And we just keep, each iteration of this loop will output a contig, and it just keeps going until we've fully depleted the database of k-mers. So relatively simple, but it's a serial process. And because of how we're selecting the seeds, it requires that we have this serial process. It's not something that could be easily made to run in parallel. We have to change something about how we're doing this. And this is the problem. We got thousands of these transcripts to assemble. Tens of thousands, hundreds of thousands, and some massive data sets. And you're going through one at a time, and it can be rather slow. So we got to figure out how can we do this in parallel. And a lot of it really boils down to where we select the seed. So if we zoom in on one of these hairy transcripts, we have a dominant path, which is shown in blue. And this just represents the path that we would get if we were doing the regular Inchworm algorithm and doing the greedy extension. And this would be the correct path, right? This would end up representing a full-length transcript sequence. But then you have all these little spurious extensions coming off of it. And these represent regions that have lower coverage. Often they're sequencing errors. And if we, in our current process, we select the most abundant seed. And the most abundant seed is very likely to end up on the dominant path, the correct path. And when we do the bidirectional extension, then we end up getting the correct transcript sequence. But we can't do this if we want to do this in parallel. With parallel, we really need to be able to seed just anywhere and start anywhere in this process. But if we choose a random seed, there is a certain likelihood that you're going to choose a k-mer that's not part of what we consider the correct path or the dominant path. Right? You could end up on one of these little extensions. And then what happens when you do the bidirectional extension? Well, you end up getting a contig which is a suboptimal contig. Right? You've got part of the dominant path, but you've also got the spurious extension here, and you end up with a fragmented transcript. So we got to figure out how to fix this. And we can show that if we do this, if we take this approach, we just do random seeding, we end up with fewer full-length transcripts. So in one of the test data sets that we use, we have 50 million Illumina reads from mouse. With our prioritized seed selection, and we look at the number of full-length transcripts we were able to reconstruct just with a simple Inchworm algorithm, we're over 7,000 transcripts reconstructed. But if we do this random seed approach, again, some fraction of the time, you're going to end up selecting something which is not an optimal seed, and you end up fragmenting the transcript, and so we get a large reduction in the number of full-length transcripts that we're able to reconstruct. So this is a problem. But one of the things that we realize is that we can just reseed from the wrong seed, all right? So I'll show this again. I like this. If we take our seed here, we build our first contig in this first round, and then instead of reporting this contig, we use this contig as a way of finding a better seed, all right? Then all we do is just look for, within that contig, what k-mer is found to be the most abundant, and that's more likely to be a better seed to start from. And so we just relocate it, find a new seed, and then we do a second round of reconstruction. Do the bidirectional extension from here, and voila, we get our correct contig. So nothing too complicated. And we can show that with our test data set, 50 million Illumina reads, by doing this, this two-phased approach, two-phase seed selection approach, we end up recovering our full-length transcript reconstruction. There's a small difference here, which hasn't been explained yet, but overall it's doing quite well. And since this two-phase seed selection actually works, we now have an option for how we can pursue a parallel assembly algorithm for Inchworm. Now, just need a few minor tweaks to our earlier pseudocode to introduce parallelization. First thing we do is we don't prioritize any of the seeds, so we can just weed out the sorting event. And the sort doesn't really take that long anyway, so it's not like we're going to get huge gains from not doing this. But it's just unnecessary. And then in our contig building phase, we build the first contig from a random k-mer, and we reseed from that contig, and build the proper contig, and report that proper contig, and then just keep going through and doing this iterative approach. And so again, this was written in C++, and it's very easy in C++ to parallelize this kind of a loop with a technology called OpenMP. You basically just add one line of code up here at the top of this while, put a pound or hash, or it's like a comment line, pound pragma OMP parallel, and you can indicate like to what extent you want it to be parallel and how the parallelization should sort of divide up the data. But it's basically just one line of code, and voila, it's multi-threaded. So it's just really awesome technology. Now, if we can look and see how well the system works, we have our original Inchworm, which is running in serial. And we have our parallel Inchworm. I'm going to call this OMP parallel. And again, we're not doing supercomputing yet, where this is all just running on like a regular server that we have in-house. And we see, again, we've got reasonable recovery, still a slight difference here that needs to be explained at some point. But then we can run this using 8 threads instead of 1 thread. And so now we're doing, each thread is doing a separate job. And we still end up with a similar full-length reconstruction capabilities. And if we look at the output of each of the threads, like which contigs they're reconstructing individually, we can see, is there a lot of overlap? Are they working on the same contigs? They're working on different contigs. And what we find is that, if we look at the contigs from each of these 8 threads, there's very little overlap. I mean, the work is basically almost equally distributed among each of the threads. And there are a few cases where they end up seeding on different parts of the same contig and end up reporting the same contig. But the redundancy is actually very, very low. All right, so this system seems to work, and as far as overall running time, the contig building running time is actually much, much faster. So here's our original Inchworm. If we just use 1 thread, then the parallel Inchworm is going to be almost twice as slow, right? Because it's doing 2 phases instead of 1 phase for doing the contig, right? For every contig, it's got to basically build it twice. So it's slower with 1 thread, but we can keep increasing the number of threads that we give it, and we can make the contig building part much, much faster. With OMP, I don't know a whole lot about this as far as like the hardware goes, and but from what I hear from my colleagues that are experts in this area is that basically with multi-threading, once you get past around like 8 threads with OMP, there's a lot of diminishing returns. All right, so usually it's around 8 threads is where you sort of get your maximum gain, and then after that it's just like small increments. It sort of saturates. But it's nice to see that, given a single server, we can still do this in parallel and achieve greater efficiency. Yes?
Don't you have a problem now with the fact that in the serial approach, you were able to remove the context that you used from the hash table?
Yeah.
Not able to use them again. But here, since everything is parallel,
Oh, they do the same thing. They still, once they've reported a config, then they delete the cameras from that config. The way you can end up with redundancy is if this is basically like a race condition, right? Where both have actually started on different parts of that config, end up reconstructing the same config, and then they're both going through the deletion process around the same time. But yeah, so it's kind of a race, but there's not really much conflict because as long as one of them gets the full thing, then you're fine. And dealing with a little bit of redundancy is not that awful. Basically, there's a final step, which is not being shown here, that basically goes through the output of each of the threads and weeds out the redundant contigs.
I assume that if you use more threads or try to count a little bit more, then you will have that problem will become.
Absolutely. Yeah. And I think I looked at that at like 100, well, we'll get to that later. We have basically a similar approach we do in the supercomputing, and I'll show you how we do that. And that's one of the things that we looked at. But I don't have a similar graph. I know that the output is very similar to like 100 processes. Once you get up to 1,000, then you start having fragmentation issues and other things that we have to do. But I haven't specifically looked at redundancy there.
Or do you have a way to detect when you've got 2 things working on something at once? Because in theory, one just stole those cameras that you would need to finish potentially building the other one. So is there like a way to backtrack in case, is there some method that detects that?
Yeah.
Those cameras back to the hash table?
Well, there are a few things that we thought about. One is just have, like, once you use a camera, immediately flag it that it's been used, all right? But then you have a problem where, if you have two working on different parts of the same config, then you're going to end up fragmenting the config. Like neither one is going to get the full-length config, right? But by not doing that, by letting each one sort of do its own thing, and even if a camera is being used at a given time, it's still fair game for other configs to use that until it's been deleted. So there, what happens is that you could have two of them. They either both get the full-length config, and then they're both deleting the cameras around the same time, which isn't a problem. Or one that's got the full-length config, and then starts deleting it, and the other one's still working on it, and it has to stop, right? Or it ends up going the wrong way. But that's less of a problem. As long as you capture, at least one of them gets the full length, then you're fine. Okay, so this is just the config building phase, but we know that within this process, there's a lot of other things that are going on. There's this initial process. We have to get all the data into RAM. This parallelization is not really helping us in that area. And that's just showing here that even though we're 14 times faster with the multi-threading and the config building stage, if we look at the entire program runtime, we're only 2 times faster because there's really these two major phases, right? You get the camera building phase, the catalog building phase, and you've got the config building. Config building gets very, very quick, but then overall runtime is really dominated by the other part that hasn't been affected by this, which is just trying to get all the data up into RAM. So.
Oh, just one more question.
Yeah.
So what happens in duplication? Like for instance, the camera should be in 2 different places.
Yeah, it's fine as long as later on in Trinity, when you have your De Bruijn graph, if the De Bruijn graph is correct, it recruits all the configs and all the bits and pieces that you need, then you're fine, all right? And so this initial phase is really just about trying to take all the reads and sort of distill them down into what are the bits and pieces that matter, and then we can.
Each is a brute force, and the rest is kind of basic.
Exactly, right. And even if we start fragmenting things here, we can still compensate for that in the later parts of Trinity. Because as long as the pieces can get glued back together again during the clustering phase, the config clustering phase, then we're golden. All right. So we still have some issues here. So OpenMP Inchworm, this is great that we now have a parallel algorithm that we can continue to work on to try to improve on scalability in other ways. But on this one big server, we're still limited to the scaling that we can get on the server, which is really around like 8 threads or so with OpenMP. And we have a limited number of cores total, and we have limited RAM, right? So the machine that we use, we got like a 512 gig RAM, which is fine for most cases, but you're confined, right? You can't really just maybe get a terabyte machine or something. I mean, there are other options, but you're always confined. You can't just add more servers or add more nodes that already exist in order to expand on the amount of RAM that you have available to you. So our goal is really to get this to run in a distributed way, so we can make use of more nodes as a way to expand the performance of the system. And so we turn to message passing interface, which is MPI programming. And in this case, basically what MPI involves is you have a bunch of servers that are connected through a network, and you can run, in this case, we'll have Inchworm. We have different instances of this program running on these different servers, right? And they're all sort of running more or less independently. And you can distribute the data across all these different machines. But then these different processes, they talk to each other. So in this case, Inchworm 1, it's great when they do this, isn't it? You have Inchworm process 1 running on this server, and it needs some data from one of the other servers that's also running Inchworm. So what it does is basically just sends this MPI send routine, send a message, give me some data, and there's a receive component, a receive method that's running over here that gets that message, and you can send very different kinds of messages. You can send messages just do this, and you're not really requiring a response back, or you're actually asking for something, and I'll have to send you something back in return. So in this case, Inchworm process 1 is sending a process 2, give me some data, and process 2 sends it back the data, and process 1 can then go on and do its Mirrorless thing. And all these processes, there's lots of ways to configure this, but in our instance, we have all the processes all communicating with each other. All right, so it's basically this big, network of communication going on. It's a big storm of communication. And one of the beauties of this process, if it does scale well, is that if you have more data, you have more need for RAM. You can just have more processes running, right? If every node has, like, say, 64 gig of RAM, and you need a terabyte of RAM, you can just add on however many nodes you need in order to fulfill that memory requirement. And in our case, we're working with Cray Supercomputers on engineering this and using their hardware for exploring this. And it's essentially the kind of system they have. It's a very powerful system. One of these has 128 nodes, two CPUs per node, 12 cores per CPU. So just one of these will give you over 3,000 cores. And each of the nodes has 64 gigs of RAM. All right. A typical Trinity job, if you have like a few hundred million reads that you want to run and assemble, it's going to require over 100 gigs of RAM. And you wouldn't be able to run Trinity on one of these nodes unless you had a really small data set. So basically, in order to run Trinity on this machine, you need to find a way that you can distribute the data among more than one node. And if you have a good system, the system scales well, then you can also improve on your performance by just scaling up to large numbers of nodes and cores. If one of these machines is not enough, you can buy a rack if you have enough money for it. It's not cheap. You have this Aries fast interconnect network that allows for very fast communication between the nodes. So there's some things. So this system, this hardware is really set up very well for this type of heavy communication among all the different nodes. If we try to do something like this here on our machines, we're going to get very, very different sort of experience. And I'll maybe say something more about that later. But anyway, there are systems like the Cray. There are other systems out there like Infiniband networks, and it's just very, very fast, and it's really tuned to these kinds of systems that are communicating.
Is there any kind of topology of the network that would minimize the need for everything being interconnected? Like have the data in certain places.
Probably, I'm sure. Yeah, I'm sure that there's like a whole spectrum of different ways of doing this, and this is sort of like the one extreme, right? This is where like everything is really highly interconnected and it's like really well tuned. Yeah. And then you can do your own like sort of homegrown thing where you get the servers, you get a special network that has very fast connection between the nodes. It's probably not going to work quite as well as this, but then you have the other situation, which is lots of independent systems that all just communicate through like a regular network, and it's not really a viable solution in some instances. In instances where you don't have tons of communication going on, you can use it more like LSF, where you basically just have your system running and you say, okay, you work on this data, and then it'll come back, and then this one will keep processing it. That's probably fine.
I didn't know if maybe you could say, oh well, this section of data can work on this section, and you sort of keep them open, but then you could use something.
Possibly. I'm sure there's lots of different scenarios, for trying to approach it. But this machine it's very powerful, and we had a good time playing with it. We had to redesign Inchworm in order to make use of MPI. It wasn't like the OMP modifications where you just put in like a single line of code and all of a sudden, woohoo, the loop is parallel. This actually had to go back and like engineer things to do MPI send, MPI receive, and really a lot of logic to deal with all the communication that's going on. So we redesigned the software, a little bit of effort. In this case, we have a single Inchworm process, and we divided it into 2 threads per process. Okay, and this is actually using OMP for this part. So with OMP, you can use P threads or use other multi-threading sort of technologies or tools. But in this case, we're using a combination of OMP. So OMP is enabling parallelization that's sort of local, right? Within the single process, you have multiple threads. And we're going to use MPI for these separate processes to then communicate with each other. So this is just one Inchworm process. We have 2 threads. We're dividing the work into a config builder thread, and we have a Kamer server thread. And it's going to do pretty much what it says, right? The config builder thread is going to build the contigs doing the greedy extension. But then you have this Kamer server thread, which is going to take care of all the communication, right, between it and the other processes that are running on other nodes. And this one process, both of these threads, the content builder thread and the camera server thread, they both have access to the same data storage, right? It's the one camera catalog that is running with that being stored as part of that process. So this is just, this is one node. It'll run. I mean, this is one process. It'll run on one node, and we can have lots of these running, right? So we can have, hundreds, thousands of these running on the Cray, and they're all talking to each other, using MPI. So now I'm gonna walk you through how it works. And in this case, we have n number of these different Inchworm processes running on different nodes. And this is our set of reads. In this case, we're going to make it even simpler. We're not going to read in the reads. We're just read in a list of k-mers and their abundance values. So in this case, here is the different 25 mers, and here are the abundance values for those k-mers. And one of the first things we're going to do is we're going to only have each process read a small part of the file, right? So we've got this huge file of maybe like 1 billion k-mers, and we're just going to divide up that file among all of the Inchworm processes. So each one only has to read in a small section, and we'll just focus on what's happening here with this one process, because all the other processes are doing exactly the same thing. All right, so with this first process, it's reading in these k-mers. And since we're not actually building contigs yet, we're gonna use this contig thread, the contig builder thread here. We're gonna use that to doing the reading, and we still have our camera server thread running too. So the contig builder is gonna read in its section of the file. It comes across this first k-mer, and it needs to know where is it gonna get stored. Right? Do I store it locally as part of my process, or do I throw it over to one of the other processes and have it store it there? This is our way of basically distributing the data across all these different nodes. And the way we figure out which node should this k-mer be stored at is just by looking at the k-mer sequence itself. And ideally, we'll have all similar k-mers being stored at the same process, right? Because when we're doing the contig extension, we always have 4 possibilities, right? Because of GATC. It'd be really useful to have all of those 4 possibilities being stored at the same location, right, the same process. So when we're doing our extensions, we only need to look for it in one place. All 4 are going to be found in the same place. We can just choose the more abundant one. The way we figure out which process stores this k-mer is just by looking at the k-mer sequence, and you basically just take the central part of the k-mer, right, missing the first base and the last base. Just take the central portion, and based on the content, based on the composition of this central k-mer, we can figure out exactly which node does this go on. You basically, the way this works, you just turn this into a number, and then you can just you run a modulus operator on it, do a mod number of nodes you have, right? And that will tell you exactly which node out of your n nodes this k-mer should be stored at. So it's kind of a neat thing. Now, in this case, this k-mer indicates that it needs to be stored at process 1, and since this is already process 1, it knows, okay, I don't have to send it anywhere, I can just store it locally. So it becomes part of its own k-mer catalog. Then we read in the second k-mer, and we look at its central k-mer sequence, figure out which node it's supposed to go to, where its destination is. In this case, it's MPI proc 2. So in this case, this process is to send this k-mer to be stored over in this process, and that's what it does. This thread, the contig builder thread, sends a message using MPI send to node 2. Node 2 receives that message, and it stores that k-mer as part of its hash table. It doesn't just send the k-mer, it tells it also how many times that k-mer is found in the data set. So you're getting both the k-mer and the abundance value. And all these nodes are all doing the same thing, right? They're all running in parallel, and they're all communicating with each other. And once the file has been read, all the k-mers will be stored at their location, and they turn gold, right? So now we're in the second phase. Each node has its set of the data. The data's been more or less equally distributed among all the different nodes, and now we need to start building the contigs. So the first thing it's going to do is pull out a seed config, and we know we can start with any seed. It doesn't have to be a priority seed. We can just start anywhere because we're going to do the same parallel algorithm where we do the two-phase search. So we just pick out any seed from our local K-mer database and at random. And we have to do the same thing where we do a greedy extension to the right. So we need to know, so this is basically, it's the same algorithm. We need to know which extension do we go to. And in order to ask that question, we need to know which process would that next K-mer exist at, right? And all of these four will hopefully be on the same node. So we just need to figure out which node is going to harbor that K-mer. And the way we do that is just look at the central K-mer for the possible extension. It tells us which process to go to. In this case, it's 2 again. I should probably change this and make it like n or 3 or something. So the config builder thread of process 1 sends a message to process 2. Can I please have the greedy extension to this K-mer? And process 2 will look up the 4 possible extensions, figure out which one is best, and send it back. This is the extension that you want. And then it goes on, and it extends it over to by 1 base, and then it just keeps going in that direction. And it's basically the same thing. We just keep going until we can't go any further. Then we turn around and go back the other way. And then you've got your K-mer, you report the K-mer. They're all doing this in parallel. So this is a nice way to sort of split up the parallelization across all these different nodes. And the rest of the process is the same too. So after process 1 reports its config, it's then going to send delete messages to remove the K-mers, not only from its local store, but from the other stores that contain that K-mer, and they all continue until they run out of K-mers. System scales reasonably well. We have the combined runtime here, as well as the time for the content building phase and the K-mer storing phase. And one thing you can see here is that once you go from 1 thread to then 2 threads, the content building phase gets a lot slower, right? And this is because with 1 thread, you're not doing any communication. You only have one process that's doing all the work, right? Once you add another process, then you get all this communication going back and forth because they're asking each other for K-mers. And but then as you start throwing in more and more MPI processes, and you've got like 3,000 nodes on this machine, you can really crank this thing up, and you just keep getting faster and faster and faster. At some point, you do end up getting to or diminishing returns, but it is much, much faster. We had reasonable scalability here. And even here, when the content building step is slower with 2 threads than is 1 thread, because these 2 nodes are splitting up the file and in terms of reading and storing the data, that process is very, very fast now. And the combined runtime is actually much, much faster. So it sort of compensates for the increased building time. So the guys at Cray were pretty happy with this, and works for me. All right, so the OMP Inchworm is just using the OMP running on a single server. We ran this on the Cray and compared it to the MPI Inchworm. And we can see the OMP Inchworm is about 3 times faster. So it's faster than it was running here at the Broad, but this is a super fast supercomputer, so you might expect that. And the MPI Inchworm is actually faster with the same number of processes using MPI instead of doing the multi-threading. But one of the key benefit here is that we're not limited to 1 node with 24 cores. We can crank this thing up, and we can just keep adding on more nodes if we need more RAM, or we want just to get the thing to just run faster altogether. So there's a couple of reasons why you'd want to do that. So we're very happy with that. The OpenMP multi-threaded parallel version of Inchworm was part of our Trinity software that we released back in April. So this is actually the current release. And it is, it's a lot faster than the earlier version. People appreciate that. We're hoping to have a full MPI Trinity, not just for Inchworm, but we've got separate efforts for using MPI to parallelize the Chrysalis step and some of the other aspects of Trinity. And we're hoping that together, it's such a combined effort between us here at the Broad, the folks at Cray, and there's a group at IBM that have been focusing on the Chrysalis MPI. And we're hoping that we can sort of combine all efforts and pull together one big Trinity MPI package that we can release later this year and show the performance, not only on the Cray, but also on other types of hardware. Now, as I said earlier, transcript reconstruction, it really is the end of the beginning. How am I doing on time here? Let's see. 1030. Okay. Oh, okay. Good. So transcript reconstruction through Trinity is really the end of the beginning. There are all these other things that we really want to support in order to really better understand the transcripts that are being reconstructed. What does it mean as far as the biology, evolution, and also being able to look at the results of doing different kinds of experiments and understanding how these different genes are being expressed under different conditions or different tissues. These are all things that interest us, and also things that we can do in the cancer domain, which I'll mention. I already mentioned the protocol paper, and this is basically one of our first efforts to try to do that, sort of go beyond transcript assembly and start enabling additional functionality. There are other things we've been working on, like the Trinity software, which is a tool for doing annotation of these transcripts. And once you have your annotations, you can do things like you run your BLAST searches, look for PFAM domains, extract the coding regions. There's other things in terms of functional categories of gene ontology. All the data is run through various compute pipelines. The results are stored in an SQLite database, and then we end up generating a final Excel spreadsheet, basically, that has all the functional annotations for your different transcripts, and that's really useful. We have another tool that's sort of extension of that, and I know there's a lot of other people here at the Broad that are doing very similar things and trying to develop tools for doing for analyzing your expression data. And there's a lot of tools that already exist out there, but it's one of those things where no one tool does everything that you need it to, and there's things that we want to do with Trinity that maybe aren't really well suited for other things. Anyway, there's a tool that we've been playing with. It's more of a toy right now. It's a useful toy, but hopefully someday it'll be more than that. Where it's a little web-based application that users can run on their own desktops. Has a little Python web server that runs. It's really lightweight. I like to build things that are really lightweight, that an individual person can do everything right there at their own computer, in addition to having other things where lots of people can work on a larger data set and interact. But this is a real lightweight system, and you take all the Trinity data that's in the SQLite database. This web-based system will query that SQLite database. You can put the expression data in there, and you can do things like look at heat maps and query genes. Put your differential expression data in there so you can look at your MA plots, volcano plots, have heat maps to look at differential expression across different conditions. And one of the things that's really useful about this is it's really exploratory, and the graphs are all, they're all very interactive, right? So you can zoom in on different regions, you can click data points. We have it set up so that you can interact with IGV. So if you find a gene that you're interested in, you click on it, and it'll bring up IGV. You can look at, here's the transcript, here's the read coverage, lots of other information that you might want to show in IGV. Looking at expression profiles for individual genes or sets of genes. Annotation reports, so we have things like the PFAM domains and the BLAST hits. Here you can look at it in this little mini genome viewer where you can see, okay, here's the transcript, here's where my open reading frame is, and here's where the domains and the different BLAST hits fall and what they are. Extract your protein sequences and your transcript sequences for doing other kinds of things. It's really a toy, and it's just something that we're playing with right now. But I'm having fun with it, and it's been useful. It doesn't really scale well right now to large data sets. I've got some massive data sets that I try to put in here as far as expression data goes, and it's not working well. So some retooling that we need to do there in order to get it to work better on larger expression data sets. Anyway, it's there. It's something that we continue to think about. As far as web graphics. I know there's a lot of interest in using D3 and all these other cool technologies. It is really exciting stuff. There's a toolkit that's it's very easy to use. I like easy. I'll say easy a lot. There's a toolkit called Canvas Express. You can go to canvasexpress.org. It's a free JavaScript library, and it gives you all these really neat plots. So you can do networks for systems biology. We'll probably use this for looking at alternative splicing in our Trinity assemblies. Lots of other sort of genome-related plots. Just lots of neat things. There's dozens and dozens of different plots, and all you need to do is just wrap your data in JSON and use their library, and it'll display the plots. And the plots are all interactive. They zoom. They do all these really cool things. You just go to the website. You can play around with all the different examples they have. And I want to just close with a little description of what we're doing and why and how we're funded. So Trinity is currently funded by ITCR, which is Informatics Technology for Cancer Research. It's a program within the NCI organization to empower NCI-funded individual cancer researchers with the tools and resources that they need in order to better study cancer. As far as RNA-seq, there's a lot of overlap between using RNA-seq and being able to study cancer. RNA-seq can essentially give you a view of the expressed exome, if you will. So you can do things like look for SNPs within exons that are expressed. It's a limited view, but it's already naturally amplified, and it's a very accessible data type, especially if you're going to do things at the single cell level, where it's more tractable than doing something like exome sequencing at single cell level. So you do the SNPs, look for gene fusions, look at transcript structures, novel transcripts that you find within cancer, look at the aberration of expression or aberrant alternative splicing isoforms, looking at intratumor variability. And there's a lot of overlap between what you can do in cancer and what the capabilities that we want to have for Trinity as a software package. So we're funded to basically provide researchers with computational resources for studying these RNA-seq related things in cancer, but in addition, to continue to build Trinity, maintain it, enhance it, and whatever we do in order to improve on its capabilities, it will be largely enjoyed and appreciated by all the folks that are working outside of the cancer domain, such as non-model organisms. And it's just a quick call out to the work that has been done recently in the Claremont Cell Observatory. This paper was just published just within the last 2 weeks, looking at single-cell RNA-seq intratumoral heterogeneity in glioblastoma. Glioblastoma is it's a very, very deadly form of brain cancer, and they basically studied RNA-seq at the single-cell level. And there's all these things. I mean, they basically looked at almost everything you could do with RNA-seq data at the single-cell level in cancer. So looking at splicing aberrations, actually oncogenic splicing variations in EGFR, finding mutations in TP53. You can actually do things. So I say copy number variation can be studied with RNA-seq. This is sort of like at the extreme level, where you can actually see full chromosome gain and chromosome loss within cancer cells at the single-cell level using RNA-seq data, which is pretty amazing. I definitely encourage you, if you're interested, just check out this paper. It's really a phenomenal work. Our goal is really to be able to empower all cancer researchers with access to these kinds of tools and being able to do these kinds of analyses, and that's what we're going to try to do, and also fold Trinity into it. And this work is going to be done in collaboration with Indiana University, where they have a supercomputing center. They have a really nice Cray machine, so it'd be really useful if we could take what we've learned with using our Cray and maybe run it on there. It remains to be seen yet whether we'll actually be able to do that or not, based on some political issues. But either way, they've got, it's a supercomputing center, and they've got lots of resources, and we're going to use Galaxy to build these great pipelines. And anyone can just upload their FASTQ files for single cell data or tissue data, and really get at the heart of cancer to the extent that you can with RNA-seq data. That's really the ultimate goal. And it's like to thank people in the group. So Aviv Regev she provides great leadership for this project. I can't really say enough good things about her. She's been just a huge asset and very supportive of my work and work of the group. Ami, I'd like to put a shout out to Ami, who's been developing the GATK best practices for RNA-seq data, and we're working with him and trying to leverage that as part of our initial offerings for mutation detection using RNA-seq and genomic data, as well as within the context of Trinity transcripts. And Tim, who was hired a couple months ago, and he volunteered me for this talk. So I want to say, if you like the talk, you can thank me. If you don't like it, you can thank Tim. So. And again, there's the folks at Cray and our Indiana University collaborators for helping with the performance evaluations and really fine-tuning the various aspects of Trinity. Thanks for coming. I appreciate it.